My name is Tom Gressner. I'm the coordinator of the European Reference Network for Rare Neurological Diseases, and also on behalf of AGI, of DGSP, and the other organizations involved in the organization of the consensus meeting on demonstrating meaningful change in HSPs and ataxias. I do welcome you to this training webinar. This training webinar is being organized with the support of the European Rare Disease Research Alliance and the Mapi Research Trust. The title of the webinar is How to Generate Meaningful Patient Outcomes in Clinical Trials, a practical guide to navigate the FDA for patients-focused drug development guidelines. Well, the webinar will take 1.5 hours, so including Q&A. And the main webinar training objective is to learn how to capture meaningful outcomes in clinical trials. From a rare disease standpoint, the trainer is Céline Desvignes-Gleizes. So Céline has been working for the non-profit organization Mapi Research Trust for the last 9 years as a patient-centered outcome measure specialist. She co-leads, together with Orphanet, the first task in Work Package 16 in ERDERA, and the ERDERA project. And this task is focused on development of a platform to support the selection of the right set of questionnaires in a trial. And she's also an instructional designer on various topics, including patient experience data. I can also introduce my co-members in the panel, which you see. So one is Matis Novcik, the current chair of the, no, not any longer, the previous chair of the Ataxia Global Initiative, and Ira Eberhardt, who is working at the AGI office in Tübingen.
So in the past 10 years, we have seen like an increase of the importance of integrating patient voice in clinical research and trials. And we have seen more and more those terms that you can see on the screen, like patient centricity, patient-centered research, patient meaningfulness. And I know that for some of you, it could be considered like buzzwords. But if they are not, because they are not associated most of the time with a thorough methodology where the patient is really at the center of all the steps of the trial design. So that's why I think for me, the extensive work that the FDA did in the past few years, resulting in those 4 guidelines, the main use is really to provide a framework to use the appropriate methodologies. It's really to strengthen the way we capture patient invoice in order to obtain robust, meaningful, generalizable, and interpretable patient input. So to better understand their disease and also to understand the treatment effect, and with the initial intention is to inform medical product development. So that's why I think it's important to have insight from this tremendous source of information to apply on your own trial. So it was interesting because 2 or 3 people in the registration form asked the question to which extent those guidelines were used and applied, which I think is a fair question, raising another question saying, why do you bother spending 1.5 hours listening about the guidelines that actually no one is using? But I think this guideline is really worth your presence and attention, and maybe there are 3 reasons why. The first one is really that those guidelines set the new standard worldwide to integrate patient voice into trials. So for drug approval process, and the context of drug approval process is really the first intention and the first field of application. However, and it's a big however, it comes with the second reason is this is not the only intention and application, because this series have an extended scope compared to the previous gold standard FDA guidelines, which was released in 2009, which was basically focused on peer claims. And now with this series, so we start from 1 guideline to now 4, The objective is really to extend the scope to patient experience and clinical meaningfulness beyond the regulatory standards of claim. So I think that's why it's also a good reason it's like it goes way beyond regulatory process. And the third reason, which could be also of a bit of big interest for you, is this this series is particularly interesting because it could help to harmonize the practice among ERNs and even within ERNs when it comes to collect patient inputs, develop new questionnaire to allow some comparison, to maybe get benefit from what other has been doing. So I think those guidelines could definitely be the standards among ERNs, while waiting also from EMA, because so far there is no EMA equivalent of those guidelines. However, they are currently working on developing a similar framework, but there is no deadline yet about when those guidelines will be released. So as Mapi said, like the objective of this training is really for you to be able to locate what is the guidelines to review when it comes to collect comprehensive and representative input from patient. So it's guideline 1. Identify the methods to understand patient experience with their disease and treatment and what matters to them. So this is guideline 2. Guideline 3, select the outcome measures that that's to be assessed to show that they are meaningful to patient. And the fourth one is to translate this measure, this questionnaire that you have into endpoint to actually assess meaningful treatment impacts. And this is the final guidelines. So before we move to dive into the proper, let's say, guidelines review, we discussed with Mathis that it could be nice to start a little bit with a terminology. We know that in our field, terminology is very important. We kept on having endless conversation on what term to use. So let's say that since we're talking about FDA guidance, we are going to use FDA guidance. It will also help you when you review them, just to have the right corresponding terms that maybe you use in your own teams. So the first important terminology, and this is the only one that we're going to spend time on it, is the star of this presentation, aka clinical outcome assessment, COA. So it's also referred as questionnaire, instrument, measure, tool, scale, test that cannot be used equivalently. However, if they are used equivalently, there are 4 types of them. So patient-reported outcome, observer-reported outcome, clinician-reported outcome, and performance-reported outcome. They are indeed reflecting different perspectives from the patient health, which is why they are not used in the same context. So I know that for some of you, those terms are quite clear, but I think it's good to have a kind of refresher on what are those questionnaires and how different they are. Because at some point, when it will come to select a questionnaire, you need to select what will be the type of questionnaire. So we need to know what are the differences. So to do so, I will display on the screen like a definition, an FDA definition, and I open the chat now, and I will invite you to tell me in the chat which COA this definition refers to by referring to the letter. So for example, if the definition is the definition of a patient-reported outcome, then you can drop the A. Okay, so let's start with the first definition. So which COA is a report coming from a trained healthcare professional using clinical judgment? Is it A, B, C, or D? C. I can see a wave of C. Amazing. Yeah, that's it. I think what is important is, yes, it's the C. Is usually the first questionnaire that has been used like 30 years ago were ClinRO, which reflect the clinician perspective on patient, which could make a difference when it's the patient answering. So next definition, reports coming, what is the COA reporting directly from the patient? Okay, amazing. Yeah, I see that you've done your homework. It's cool. So yeah, it's definitely the PROs. Here we have a Q5D and SF36, which are like gold standard of PROs. So this one, which provides a report coming from someone other than a patient or healthcare professional. Yeah, I can see the B already. Here, I just will pause 1 second on this one, because this is very of importance, particularly for the rare disease field, because OPSRO can be used for two populations, old population, like population with mild cognitive impairment, like Alzheimer, Parkinson's disease, and also be used for pediatric population, like the PET score. The PET score is the gold standard for quality of life assessment in pediatric population. We know that there is a scarcity in OPSRO, and there are some challenges, but to turn PROs into OPSRO in order to make one scale to fit this population, but we'll see how to do that later on in the presentation. So the final one is a bit aside, I would say, is the performance outcome, which is a measurement based on a standardized task. So it could be walking, could be sitting up and down, could be also cognitive tests. So altogether, you can see they do not assess the same thing. It's like there could be like the clinician perspective on patient health, caregiver on patient health, patient on him or herself, and this task performance. So let's have now a high-level review of what those 4 guidelines are. So the first one basically has been finalized in 2018 and focus on how to collect comprehensive and representative inputs. So basically focusing on the population, so sampling and representativeness of the population. And it also raised the importance of setting the trial objective right at the beginning, the research question that the trial aims at addressing. The second has been finalized in 2020 and 2022 and focused on the method to identify what is important for the patients. So here there is really a focus on how to collect the meaningful aspect of health of a patient. The third one focus on how to select, develop, or modify fit-for-purpose COAs. So here the key driver is really COAs. So it's the one these guidelines will spend the most time on. And the fourth one is about on how to incorporate COAs into endpoints for regulatory decision making. So here the key point is really this: how you turn patient voice, patient verbatim into something in the end that is quantifiable and comparable. This guidelines, same, I will not go through in much details because you will have like a proper meeting on that later this week. So this will be our framework. And as you can see, the framework is actually the different steps to build a patient-centered trial, a trial design. So that's why I think it makes also sense to do this way, like to review from 1 to 4. They make sense as well, chronologically speaking. So let's start with the first one. So in order to have like a frame, each guidelines will be described the same way with the objectives, the key recommendation, key notions as well, and also what it means for rare disease field, as much as we can. So the first, I mean, the objective of the first one is the importance of defining your trial objective. What is your research question you want to address? To determine the target population, discuss sampling methods to ensure representativeness, and also to perform like a background review to identify what would be the best way to collect patient input, qualitative method, quantitative method, a mix of the two. So basically the key recommendation is for this guideline 1 is perform a background research to identify the trial objective, patient population, methods to determine what is important for the patient. These guidelines, I mean, we're going to review it quite quickly, but this key notion of patient experience data is very important because it's the very first time that FDA introduced this definition, and it really reflects the fact what I start with is like those guidelines are way more than just choosing a questionnaire. is how do we collect patient experience? And patient experience is quite broad because it's the information that capture patient experience, perspective, need, and priorities. And those, for example, those priorities needs to be also reflected in the endpoint, like the primary endpoint versus something vital or something of huge need for patient compared to secondary endpoint, for example. So the patient experience data encompasses symptoms, the impact of the condition on functioning, quality of life, experience with treatments as well. Could be ease of use, for example, is the treatment easy to administer, for example, or which outcome are important. And something also new is like the patient experience preference for outcome and treatments. Now in this patient experience data, preference studies are included as part of patient experience. So if the administration route of a certain therapy could be assessed and could be considered as patient experience data, because preference of how the drug could be delivered, for example, is of importance now. So what it means for rare disease. In this section for the 4 guidelines, I also implemented data from a very recent guidelines that has been released like 1.5 years ago on the consideration for drug development and biological products for rare diseases. So I really encourage you as well to have a look at this one. But I also extract from these guidelines information that could be useful to interpret what is reported in the PFDD guidance. So what we can say is not something super new, right? It's just to find the right balance between constraining patient eligibility to decrease the heterogeneity and also to find the right balance with inclusion of a broad range of disease severity, stages, phenotypes to ensure representativeness. So it's subtle balance, it's true. But what is highlighted in the guidelines is that further later, when it will come to assess the validity, sensitivity, reliability, and interpretability of the tool that you're going to use for the endpoints, they may be different for patients if they are mild, early stage, slowly progressive forms compared to patients with more severe late stage or rapidly progressive forms. The threshold of meaningfulness, for example, could be different. So it's how do we deal with this when it comes to sampling or to inclusion criteria, basically. So this is it for the first guidelines. So here the key notion is the patient experience data, what it means, how broad the scope is. For the guideline 2, we are going to to see here, so the objective is, key point is methods. It's really a methodology guideline to identify what matters the most to patient and identify meaningful aspects of health that are impacted by the disease. So the key recommendation is, first, whatever method you apply it thoughtfully and with a proactive rationale. Is if you use this method, why you use this method. Basically, there is no perfect method. It's just why you're using it and apply it thoughtfully. So these guidelines describe 4 methods. So qualitative methods with interviews and focus group, quantitative methods with surveys, mixed method research, which is a blend of the 2 first, and social media. I'm just going to spend time a little bit more on quali methods and mixed method research. But just a few notes about social media, because it's quite new. However, it's worth mentioning that particularly for rare disease could be very helpful. FDA is more open to consider information from social media. Should be carefully, the source should be carefully selected. And also the data analysis should address potential limitation, like the difficulty to track the patient characteristics and how they can affect the data integrity and interpretation. It's also kind of straightforward, but however, it's interesting to see that social media could complement information coming from the 3 other methods. So for qualitative method, the objective, what has been in phases, is like to have unbiased patient input. So to use a semi-structured interview guide, not suggesting any answer, not assuming to know what the participant is thinking or feeling, and not asking questions that cast judgment or imply what will be your preference over patient's preference. Same, kind of obvious, but the conclusion that FDA highlighted is like, it's important to invest in the training of interviewers to ensure the fact that the way the interview would be performed is less biased as possible. Here, I just wanted also to emphasize on this, what we call MMR, mixed method research, because this is definitely something that we see increasing, particularly in rare disease. And you're going to see why. I've described like 4 scenarios just to picture how we can have this blending of methods. Because the objective of this MMR is to supplement or clarify results that could come either for quali method or from surveys. So let's have an example. So you can perform a focus group to have a broad insight of which way the disease impacts the patient's life. And then you can have like one-to-one interviews to have a deeper understanding, either on one maybe stages of the disease or one part of the population, like female or man, or like more early stage of the disease, to have a deeper understanding of how the disease impacts. Particularly because of the heterogeneity of the disease, this combination of those 2 qualitative methods could be interesting. Second way to have this mixed method research is here is an example on exploring depression and anxiety in patients with acute coronary syndromes. It's just here an example for the sake of the presentation. So you have like a survey to apply, a questionnaire. You analyze the survey, and then the result of the analysis shows that there is a correlation between depression and anxiety with females. So you perform a one-on-one interviews to explore a bit more why there is such relationships. Another aspect is to perform in parallel surveys here to explore symptoms, for example, with a checklist, and you also ask the patient to describe their symptoms of the disease. You apply analysis for both methods, and then you allow comparison of those analyses to just see how they can supplement or clarify one method over the others. And the final one, I think it's also something growing, is the use of exit interviews. So for example, in a trial, you explore functional impacts, and at the end of the trial, you ask the patient in an exit interview, what was the impact of the treatment? It could be broad questions, but it allows you to confirm that what you assess with the questionnaire was actually a good questionnaire. But sometimes we do not always know which extent the treatment benefits. And this exit interview can also help to identify impacts that you did not anticipate, that now you'll know with these exit interviews, and therefore for the next trial, you will include those concepts, those impacts into a questionnaire. So it really helps to make sure that you get a large, exhaustive sense of the treatment impact and the disease impact. So whatever you're going to do, what is very important is to have the rationale behind this. It's not, oh, I will try to do this, or let's see. I think it's important to already have in mind why you're doing it. So I also go back again to the same thing. What is your objective? What's your trial objective? And one of the key information from the PFDD guidance is really, how about interpretation? Interpretation of data is key. That's why it's important to anticipate, if you're using both quali and quanti research component, is that you clarify in advance how you're going to interpret the results. The key notion of the guidance too here is this is conceptual model. I'm not a fan of all theory and notions, so I introduce only the one that will be helpful for you, and you will see it after why it's important to build this conceptual model. Conceptual model, this is a very fancy definition, but here is really to propose causal linkage among a set of concepts believed to be related to a peculiar disease. Here it is. is basically a concept that are arranged according to different categories as you needed. So it could be like signs and symptoms, disease management, impacts on functioning, treatment effects. And there could be simple, there could be complex, there could be categories, subcategories. It's really up to the condition and also what's the objective of drawing a disease conceptual model. This is an example of ataxia. So we shorten it, we sampled it because obviously we have way more information, but here it's an example of sign and symptoms. You can see 3 symptoms that has been highlighted by the patient on the purple parts: the impact on quality of life, on activity of daily living, physical, emotional, social functioning, on sleep as well. Also aspect on disease management, on the treatment, on the decision-making process, and how was the healthcare management. So all that together, it's just to make something visual out of patient verbatims and how to see how all those things are connected, and also what is important, because maybe you have identified 4 publications and the 4 publications mention that there is a huge impact on activity of daily living and only 1 on sleep. So maybe in terms of priorities of importance for patient, ADL will be maybe more important for patient to be improved than sleep, for example. It's just an example for the sake of the presentation. So what it means for rare disease. So because of a limited understanding of natural history that we could have sometimes for some rare disease, a lack of regulatory precedent, the rare disease heterogeneity must be documented, and to really efficiently assess what will be the treatment benefit. If we assess if we identify the right meaningful aspect of health, then it will be easier to assess the efficacy of the treatment for each stage and each level of severity. And basically what FDA recommends is like to blend methods to capture a robust and comprehensive understanding of patient experience. Okay, so there it is for the second one, which I think here, the key concept is this visual way of seeing the impact of the disease on patient lives. So altogether, those 2 guidelines really help to, this is really the starting point. It's very, even if we know that we're going to be way more interested in the third one, what's important with the first and the second is like they provide you with methods to obtain robust, meaningful, and interpretable patient input to really understand what is their experience of their disease and the treatment. Do we have question in the chat, Mattis? I don't have the chat.
No, there's no chat. There's no question in the chat.
Okay. So let's dive into the third one, the big beast. So for the third one, the objectives of these guidelines is twofold. First is what to measure by identifying concept of interest to assess and adequate questionnaires. Second is how do I do that? How I select, develop, modify a fit-for-purpose COA for its context of use. Here, many terms. So the one in blue will dive into what are they precisely. So first, let's start with what to measure. Identify concept of interest and adequate COAs. So first, what is a concept of interest? So concept of interest is an aspect of an individual experience that the questionnaire is intended to capture. 3 components of a concept of interest. It should reflect the meaningful aspect of health, and this is where it's connected to the PFDD2. Second aspect important, it has to be the ability to be modified by the treatment, because maybe the treatment is not going to impact all meaningful aspect of health of patients. And the third one, it should demonstrate a clinically meaningful difference within the trial timeframe, because maybe some meaningful aspect of health do not evolve very quickly. So in the time frame of the trial, it's just not possible to see a difference. So below is example of concept of interest, symptoms, impacts, or functions. So how do we identify those concepts? First, the first starting point is really a literature review. So basically to have a review of studies of natural history, trial that have been performed in this therapeutic indication, ct.gov, PubMed, and also to review labels of drugs that have similar mechanism of action, just to see what has been done by other drug developers, what are the questionnaire they have been using, what concept they assessed. So when you have this list of concept of interest, then you get a sense of the anticipated benefit of the treatment in what we call the TPP, target product profile, which I guess when you have the trial, you have a pretty good sense of what would be the treatment effect. If it's not the case, then you can, based on the literature review, see what are the anticipated benefit and perform at the end of the trial and in exit interviews to see if there were other effects that were not anticipated. These two pieces help you to draft two lists. First, concept of interest. Second, a preliminary list of COAs that has been used in the conditions in previous trial with the same condition. Usually in your, let's say your trial structure, design structure, you may have 3 types of questionnaires. What we call generic or domain-specific, like a Q5D, SF36, or domain-specific, like sleep, fatigue questionnaire. Second, disease-specific questionnaire. And third, questionnaire to use as anchor for the assessment of meaningfulness. So based on this preliminary list, you will confirm what you have identified by performing patient interviews. So it's what we call in our own jargon concept elicitation. And concept elicitation will help you to confirm the concept of interest that you have identified in the literature review and maybe identify a new one. And will help you to finalize your list of questionnaires by performing what we call a concept coverage. So here it's just an example in myelofibrosis of how do we we select COAs based on their concept. So here it's just an example we've taken from a real-world evidence case. So here from the interviews and literature review, we have identified 6 general concept of interest. And if we apply a concept coverage and based on the also literature review, we have identified that the modified MFSAF version 2 could be a good questionnaire because it covers largely almost all the concept except fatigue. And then we have seen the PROMIS could be also a good fit. There were other questionnaire that has been assessed in fatigue, but PROMIS fatigue, what makes us take this one, or the team takes this one is the fact that he has a precedent by the FDA. It has been claimed by FDA over other questionnaires that assess fatigue. So that's why we, based on our literature review of competitors, we have decided to go with the PROMIS fatigue. So now that we know, based on the literature review, treatment effects, plus interviews, we have a rough idea of what we want to assess, what will be what are the questionnaires that exist and are available. Now we're going through a bit more in details on how to really select those questionnaires and how do we know that we need to develop a new one. So to address this objective, FDA has developed what they call a roadmap to support the decision process on the best COA to select or to develop in order to generate the evidence to address their your trial objectives. So let's have a look step by step. So first is to understand the natural history of the disease based on literature review, patient interview, caregiver interview, clinician interviews as well. Then here, we'll get a sense of what are the concepts of interest and also to define the context of use. We'll talk a bit more about context of use in a few slides. So here at this point, I just make a little stop based on question that I also received. Here is, we are talking about COA FDA roadmap. But here we have two options, or three, even more. When you know, okay, I know what is important for my patient, I know where the treatment will affect or improve my patient health, I know what I want to assess. Here I have three options, basically. I can use a biomarker to assess one of the aspects. I can use a questionnaire, but I can also use what we call now digital health technology, like actigraphy, podometer, Fitbit, biosensors. So those new tools could be also interesting because they could be good complementary. They could complement COA data, as long as you explain how they could complement and how the data can be compared or supplement. So this was just a little side note. So we go back to the COA. So we know what we want to assess, what's the target population. Based on the target population, we'll select the right type of COA, PRO, OPSRO, CleanRO, or PERFO. Then we'll search what are the existing questionnaires, basically from a literature review. And here we have three options. The first one is the best one, the gold star one, is like you have the COA that has been developed for your target population, do not need any modification. Option two, there is a COA, but it requires some modification. So when I say modification, it could be adding an item, deleting an item, modifying an item. So number of response option, the type of scales. So here you need to modify the questionnaire. And the third option is like, okay, I need to address 7 concepts of interest, but in fact, no questionnaire largely cover them. So I need to make up my own questionnaire. The final goal is to show that the questionnaire that you have selected with the data that you will generate in your phase 3 trial is to show that your COA is fit for purpose. What's fit for purpose mean? Major key notion. So COA is considered fit for purpose when the level of validation associated with a medical product development tool, so COA, is sufficient to support its context of use. So basically, to make sure that COA is fit for purpose, you need to show that it's shown evidence of a strong validation. Validation could be broken down into 2 aspects. First is what we call the content validity, using qualitative methods. So basically, interviews, we have seen that a questionnaire, if you develop a new questionnaire or if you modify one, you have to show that you have performed interviews to identify the right concept. And you also make sure with the cognitive interviews that the questionnaire basically is understood, is comprehensive, it's relevant. The recall period is appropriate. You collect the time to complete and the appropriateness of individual items. Second aspect of validation, which come after, is the psychometric properties assessment, which is based on quantitative methods, basically showing the reliability, construct validities, diversion, conversion validity, and non-group validity. The ability to detect change, that we also call responsiveness, and the establishment of a meaningful change threshold. I'm not diving into details here because that's not the purpose, but just to remind you that to show that the questionnaire is valid requires both qualitative methods and quantitative methods. And those validation steps needs to be done in the context of use. So it's basically a full clarification on what is the context in which a COA is going to be used and why, what's the purpose of its use. So it's basically to describe the target population, what would be the trial design, what would be the positioning of the COA that you're going to use, and how the COA is going to be collected, who is going to collect it and when. So altogether, this is the evidence generated to show that the questionnaire is valid, content-wise and psychometric-wise, will show that your questionnaire is actually fit for purpose. So I like to have like examples. I didn't find like a real world example that we have in rare disease that was that clear. So I took one example in celiac disease that we worked on to show you to put that all together, what we have learned. So what are the different paths, how we apply this selection modification process? So first, we have performed a literature review. We reviewed qualitative study to identify concept of interest. We also perform a literature review to identify a set of PROs that could match this concept by concept coverage, concept mapping. And based on this literature review, we have identified a preliminary set of PROs. So first, the SF12 to assess concept of interest related to health-related quality of life. Then we had 2 questionnaires to cover more disease-specific aspect. So first, symptom diary, covering important symptoms using a severity response scale, and also the ICD-SQ, the impact of celiac disease symptom questionnaires to assess other impacts. So we start with this preliminary set based on literature review. Then to confirm what we have performed, we wanted to talk with patients to ensure that we are actually covering what is really meaningful for patients. So we perform patient interviews, so what we call concept elicitation. And here are the feedback that we received from the patient. So first, they were saying that in the diary, the 2 items, diarrhea and vomiting, were used by frequency instead of severity. So in terms of response scales, they prefer to report it as frequencies instead of severity. They also mentioned new symptoms like skin problem, gas, and headaches. And they also confirmed that the items covered in the ICD-SQ were relevant, were actually important impact, reflecting important impact. So because of this frequency severity type of scale, we contacted the developer of this questionnaire, this diary, to ask if we could modify the type of response. And no modification was allowed. So we decided to develop a new PRO. So in conclusion, here is after following the different paths. So using questionnaire as is, first option of the FDA roadmap, we use the SF12 to assess health-related quality of life concept. We kept the ICD-SQ to cover disease-specific impacts, and we develop a new PRO to assess the following symptoms. First, we kept the symptoms assessed by the diary, but changed the scales for 2 of them, diarrhea and vomiting. We changed it from severity to frequency. And since based on the interview, we have highlighted like 3 new concepts were not covered by the previous diary, we included them into the new PRO. So this is in the end what we have done. So it's basically the first option and the third one. I also just wanted to, since in this example, we discussed like how to develop a new questionnaire. This is not in the PFDD guidelines, but it's more like a reminder of the steps to do when we want to develop a new COA. So the first step is the item generation based on literature review, on interviews, patient, caregiver, clinicians. Then we develop a conceptual model, as we've seen before. And then you have like a preliminary list of items. You perform preliminary questionnaire tests. So basically what we discussed before, it's part of the content validation of the questionnaire, cognitive interviews to make sure that the items are clear, they're understandable, they mean what they want to assess. And we also perform statistical analysis to make sure that there is no redundancy among different items. So we perform, for example, confirmatory factor analysis, CFA. And then after different modification, number of item, response option, recall period, we end up with the final questionnaire. And this is where we, at the final round, psychometric properties assessment with the different statistical tests that we perform to show reliability, validity, responsiveness, meaningful change. Basically, we do that. The item 1 is phase 1, with the cognitive interviews in phase 2, and in phase 3, we perform the psychometric assessment of your questionnaire. Okay, maybe I will yes, maybe we can I will just go through the roadmap to also in phases with this example, and then we'll address some questions.
That's fine. Yeah.
It's really come together. So basically what the FDA show is what here we have illustrated with examples, but FDA also provide very interesting examples on how to go through this roadmap, how to practically apply it with what they call the conceptual framework. So here is just an example of the conceptual framework. Is the first phase based on PFDD1 guidance is to design your target population. Then you perform qualitative studies with patient, caregiver, or clinical expertise to identify meaningful aspects of health. And then all those concepts together, here is feelings, for example, 5 feelings and 4 activities. You gather them into a general concept. Using here, you can also see that it's like a disease conceptual model that you are drafting. You can, here it's the upper part of the disease impact model with 1 or more concept of interest to select for assessment. Then you select the type of COA. So here, 2 PROs to assess symptoms and to assess function. And you generate score, because as we said, the score allows the interpretation, and it's what's key in the end to generate. So what it means for rare disease. There is a specific paragraph in PFDD3 guidance on how to select, develop in pediatric tool. So what they say is, if a COA can be measured across the age spectrum, it's better to favor 1 COA for all the ages. And if it's not possible, and you have different versions of the questionnaire, make sure that in the statistical analysis plan, you can reflect in the analysis the data comparison between the different age categories. They favor self-report for COA when it's feasible. Obviously, it's better without any assistance from caregiver or investigator or anyone else to avoid influencing the child answer. And they also mentioned that to have a computerized computer administration COA may ease the process of self-report. This could be one example of modification of a questionnaire to try to use simplified age-appropriate response scales, for example, with less response option or easier to get, or with pictorial scales with faces. And also, if you use, for example, a PRO that you would like to turn into an OPS row, it's important to generate evidence to show that the COA is actually suitable for pediatric population. So basically, cognitive interview, just to make sure that the patient actually understands what they're reading. And that's it for the third one. It's a big piece, and I see that there are questions in the chat, so let's go with the question in the chat.
Yes, Céline, thank you for wrapping this complex guidance up so nicely. There are 2 questions, and there were actually also a question from my side. At the very early part of your presentation of guidance number 3, you said that digital health technologies might support or complement COAs. However, and just for general disclosure, is for us in particular in a texture and HSP world, digital health technologies, in particular like body-worn sensors, are of a lot of importance. So this is why this question came up. However, we currently so far think about digital health technologies assessing, for example, mobility as a type of perfo, as a performance-related outcome, and thus as a subsection of COA. So not complementing COA, but as a type of a COA.
Yes, it's a current question that we have internally as well, because some could be considered as perfo, some can be considered as biomarker, for example. So there is no clear cut, because also DHTs can be also considered as a way of administrating a questionnaire or a perfo. So there is no clear cut. But however, I think here what's important is whatever tool you use is to provide a rationale of why you're using it, what is the concept, when you generate the data, how you're going to interpret that, particularly for DHTs, because the problem here is like usually you have a huge amount of data, and it's not always easy to find a way to interpret them.
Yeah, and I think, and thank you, Céline. I think that's a take-home message also for our Heidelberg meeting coming up, is that not just because it's a digital health company, digital health technology, it not automatically counts as a digital perfo and such as a CRA, but look at it, and we look at it and provide justification, and then it's via the justification rather than via the nature of the technology or assessment. And I think that's a take-home point. Thank you. The second point is Rutang Shah is asking, how many times do you need to conduct patient interviews? I thought we already had patient interviews part of the concept of interest. So maybe we can put this question, make this very specific. As part of your generation of the pro for celiac disease, and if you don't mind, you can go back to that slide. There you had the patient interviews as part on that. And my question then, in order to make this question more specific, is what was the function there? Did you use did it have the function to elicit a concept of interest, or did it have the function to validate the COA? So just starting off with that example which you had there.
Yeah. This one.
That one. Yeah.
Okay. So here, the first patient interviews, the objective is really to collect like the interviews that you're performing to better understand the patient. So it's basically there are two types of patient interviews. There, the patient interview here we called concept elicitation, and the second one is cognitive interview, just in case to make sure that the questionnaire is used. This first type, concept elicitation, the objective is to have a clear picture of the impact of the disease. And they are part of if you want to say a questionnaire is valid, you need to perform this, because it means that your questionnaire is based on actually what's happening for the patient. And this is crucial to show that you have literature review that could have a draft of what the disease impact model, how the disease impact the patient life, but we need to face it, to confront it with patient interviews. And this is part of the validation. When we say that the questionnaire is patient-centered, validated, this part should be done. I said patient, but it's like broadly patient or caregivers plus clinicians, ideally.
Yeah, thank you. So Rutang, the question is not so much about times, but what is COA you're developing, and at which state you're in developing, and do you need it either for concept elicitation and or for as a cognitive interview to validate your COA? And if you need to do it several times, you need to do it several times. So there's no, it doesn't go by number, but by a role in the process. Yep.
Usually it's like phase one, you have your patient interviews, then you select your questionnaires. You will need, except if you already have all the questionnaire that you need, but it's likely that you will either develop a diary or modify to your population. So as long as you change the original questionnaire, you need to make sure that what you've done is comprehensive, it's understandable by patient. And so that's why you need to, as long as you modify the questionnaire or develop a new one, you need to perform cognitive interview. But if you use the questionnaire as is, there is no need for cognitive interviews.
Okay. Céline, there is a threefold question, which is quite complex in the chat. I don't know how much time we have for more question, whether we can get into this or not. So
which one it is?
Let's see with the first one. The first, I'm just reading out loud. The disease conceptual model in terms of ataxia is to be understood as a phenotype conceptual model, correct? I'm not sure what a phenotype conceptual model is, though.
Yeah.
So maybe, Tanya, you can add to this, and while you write this down, we focus on question number 2. The second independent question, is content validation still needed in case of a specific pro has already been developed along the patient-focused drug development guidelines? I think the answer is, if it has already been developed along the patient-focused drug development guidelines, content validation has already been part of it. Otherwise, it would not have been part. So it's a little bit a circular question, right? And so okay.
If it's always the same, it needs to be done in the trial target population. It has been done for in another population. You have to perform it again just to confirm, because it's not the same population, so the impact of the disease could be different as well.
Her third question is there a space for expert consideration in selecting a concept of interest for a specific concept of use according to the expected or proven responsiveness?
Sure.
Mm-hmm.
Yes. Clinical expertise is really part of this qualitative first part here, like could be patient interview and clinician interviews. The only thing is patient needs to be involved, I would say. Then it's for a clean row, it's a bit different as well. It's really a balance of, if it's for patient-reported outcome, for sure, we need patient. This is absolutely necessary, and to reach a saturation. For clean row, I think anyway, if we want this to be patient-centered, we also need to ask the clinician, but also the patient, just also to confront this.
Right. And I also think you had one of your earlier slides when you had the combination of a literature review plus treatment anticipated benefits. Those two are the sources to generate a preliminary list of concept of interest. And this then feeds into the patient interviews to elicit for concept elicitation. And I think for this literature review plus the treatment anticipated benefits, there is a genuine role for experts. So I would not know who else would do this if not an expert, right? So I think that's nothing that cannot be done by an expert. Yeah.
Sure.
Yeah. Okay. Thank you, Céline. That were all questions from the chat.
Ooh, okay. Amazing. Thank you for your questions. Okay, there we go. So this was the big piece, right? It's a big piece, but I think it's helpful, I think, to see that patient needs to be at the center, and also the clinician. It really works. It's for me, it's kind of intuitive the way it works. Okay, so for the third one, we have seen that there are those 4 key notion: context, concept of interest, context of use, what is fit for purpose, and the FDA roadmap. So for the next guideline, so here I'm not going to spend time just high level, just to get a sense to kind of finalize, wrap up the entire process. 2 objectives is, how do I turn a list of COA, because it's what we aim at having at the end of PFDD3, and how do I turn it into actionable, quantifiable data to compare, so into an endpoint? And how do we show meaningfulness? I'm not going to talk about showing meaningfulness because it will be covered later on, but I'm going to spend time on the first one. So first, what is an endpoint? So it's an outcome that is statistically analyzed to determine the efficacy of a therapy. Great. Let's have an example. Okay, so here I carry on with the myelofibrosis example that we had for the concept coverage piece. This is the endpoint they ended up with after the concept coverage, interviews, and selection. So the primary endpoint is like a biomarker. So it's 35% reduction in spleen volume assessed by MRI at week 24 from baseline. And the second endpoint, so it's about MF symptoms improvement with a proportion of patients with more 50% reduction in MF total score from baseline to week 24. And the second part is change in fatigue in the total score of PROMIS fatigue 7-item short form from baseline to 24. So just to get a sense of what do we need to develop an endpoint. So first we need a concept of interest. So here we've seen spleen volume, MF symptoms, and fatigue. We need a tool, so a COA or a biomarker, MRI, MSF version 2, modified version, and the PROMIS fatigue. We need a score to turn this concept into something quantifiable. So a 35% reduction or total score for both questionnaires. Could be also domain score sometimes. And the timing of assessment, so from baseline to week 24 for the 3 trials. So let's say that as a conclusion, basically an endpoint needs a concept to assess, a tool to assess it, and this is here for the tool that we go to DHT, biomarker, or COA, a score or data to generate, and the timing of assessment. Maybe another example directly from the FDA here to assess like nausea. So from the patient voice to really the endpoints. The COA that has been used to assess this is nausea daily diary. The COA score here is the severity rating. And the endpoint in the end is a 7-day average nausea severity rating at 3 months post-randomization. So this second example really shows how we progress from the patient feedback to quantifiable data into an endpoint. So the key recommendation, I think the major one is this one. When we consider a patient endpoint strategy, patient-centered endpoint, it means that it needs to be both statistically significant and clinically meaningful. We need to go beyond the stats. We need to show that it's clinically meaningful. And a major part of the guideline will show how to do that with anchor-based methods. So for some condition, particularly here for rare disease, it's the case, some condition may have variable manifestation, relapsing or remitting condition. They could be progressive condition or episodic or acute condition. The objective is to anticipate, it's always the same, to anticipate this complexity by developing a statistical analysis plan early in the process with statistician to make sure that you actually capture all these complexities, and therefore you will be able to assess them and to interpret the data variation because of this complexity. And there is one important notion is it's absolutely possible, and it's likely to happen to that you will have missing data in your data collection, and also intercurrent event, which is basically what could impact the data collection, like treatment discontinuation, treatment interruption, or death. And FDA really emphasizes the fact that in your statistical analysis plan, you need to really anticipate this. Means that if one item in the promise fatigue is missed, how I'm going to deal with the score, for example. And it's really important to anticipate that, to relate to one of the key messages of the series, which is interpretation, to ensure the quality of interpretation. So here, what it means for rare disease, 4 notions. The first is like when you have both pediatric and adult participants, it's important to consider the relevance of the endpoint for both groups, and if it's possible to combine the results of the both to have one unique endpoint result in the statistical analysis plan. So in the rare disease studies, we know that it's difficult to have like a perfect endpoint. It's not always available. That's why it's very important to include the patient and the caregiver for frequency of assessment, for the selection, the development, and the modification of existing COA to improve the chance of success for the development program. So for this one, I'm not going to move with more detail. It's just to, I think the key, there is no particular key notion except the endpoint, but I think it's important that this COA-based endpoint needs to generate evidence statistically significant and also clinically meaningful. And one point is like, we could think that patient meaningfulness will be only assessed with the final guidelines, the PFDD4, with anchor-based methods, with their threshold for meaningfulness. But what I think here, what we can see now is like the patient is at all stages, from the sampling, the representativeness, from the identification of what mattered to them, for the selection of concept of interest, for the selection of COA that actually reflect what where they want the treatment to have an impact, and also then with the threshold for meaningfulness. But it really goes at every step of the trial design. Okay, so let's have the takeaway, and then we'll ask we address the last question. One single objective of this series to really emphasize the fact that we need to better understand and integrate patient voice in trials. And for that, we need to apply thorough and scientifically sound methods. Patient experience, yeah, that's something also that FDA kept on repeating. Patient experience before the measure. Clinical meaningfulness to integrate from the beginning of the trial. And one very important point is like there is no perfect endpoints. So even if we have questions on like, should I try this or this? I'm not sure I can even answer. Because what's important and what's FDA, I think it's quite encouraging and said that you need to justify why you do that. And usually it's based on patient interviews or caregiver interviews or clinician interviews to really justify what is your endpoint strategy and what is your context of use, so your target population. And this is the big part, is interpretation is key, and it's important to anticipate that when you draft your endpoint strategy. Source of information here, so the first point is the full PFDD guidance. There are 2 guidelines, very recent one on rare disease that I really encourage you to read. Some around the natural history studies, if you have specific question on it, not that old. There is one saying very recent on DHTs, if you want to review it as well. There is one on biomarker, because there was one question on biomarker that I will review just after. And this is the latest, the guidelines released by the FDA before the PFDD guidance, that it's still applicable, but here it's more for PRO claim. So we reached the end. I just would like, if you're okay, Mathis, that I review this question that you sent me.
Yeah, go ahead.
There was a question about how to anchor a surrogate, a biomarker. So what is needed for anchoring an outcome change of a COA versus a surrogate in a patient meaningfulness that it's regulatory acceptable. So we'll just display the first answer. So usually what we do to anchor, and you'll see that we use like a PGIS or a CGIS, depending on the questionnaire we use to compare to use as a COA anchoring. In theory, we could have a biomarker, another biomarker as an anchor for a biomarker. But from our experience, we have no precedent on that. In practice, we usually use the anchor, the PGIS or the CGIS, to validate the biomarker itself as an anchor. There is no, so when it comes to biomarker, there is no need of additional evidence for biomarker than for COA. I mean, for COA, it's already quite exhaustive process. And there is one guidance specific to biomarker that you can review. And that's it. We can address the rest of the questions.
Céline, thank you so much for this nice presentation. And I mean, you really covered a lot of ground and presented in a very illustrative fashion. Thanks for that. May I just start off the discussion with coming back to your very last Q&A point, just to make sure and I think that's a question many of us in the field have it, whether coming from the imaging or molecular biomarker world or digital biomarker world, where they do not qualify as a digital perfo. So if I understand correctly, actually, there is no difference between a surrogate biomarker versus a COA. Because you say, okay, pretty much you do an anchoring strategy as you would do for a COA, for example, do an anchorage in a PGIS or PGIC. You would do the same with the biomarker and no additional evidence would be needed. Do I get it right?
Yeah, I think it's usually what we do is like, even if for a biomarker, we don't go through a full validation process as we do for COA. You need just to justify why you are using this biomarker based on same background, literature review, interviews, and all of that. But when it comes to showing the meaningfulness or the interpretation of data of a biomarker, you can use the anchor that you would use also for COA. It's usually what we do. We have an example where we have the PGIS with a PRO and a biomarker, and we assess meaningfulness based on with this anchor for all the other, for the 2 PROs that we have and 1 biomarker.
Okay, thank you. Then I can start off with some question from the chat. The last question is, we can refer to Wednesday. Rutan, your question is, how can we demonstrate clinical meaningfulness when there's no available treatment? I think that's a discussion we can focus more in depth on Wednesday. But for sure, Rutan, just to let you know, you don't have to have an actual treatment available in order to be able to show clinical meaningfulness. So that is you use those anchor-based measures where you show that a certain delta change in the clinical outcome is related to a certain delta change in a certain anchor variable, and for that you don't need to have a treatment at hand already. But that is we focus on more depths in Wednesday. There's one question from Fehork in the chat, Céline, and that is a question: if you aim to have a COA not for a disease, but for a specific sign, so a sign as opposed to a disease, and the sign let be, for example, imbalance, rather than for a disease group like ataxias, how do we then draw a disease concept? Because you have been emphasizing the need for a disease concept, but how about if not disease is a focus, but a sign is a focus? Or can you do so at all according to the FDA guidance? Or would they always say, don't stick with science, anchor it, or use it in the context of a disease in order to make it fit for purpose?
So the question is about the disease conceptual model. Do you need that if you focus only on one concept?
If you focus only on a sign as opposed to a disease, do we then still need a disease model? So let's assume we have a COA. Let's assume we have a gait score or digital wearable for imbalance, measuring stumbling, strange gait, but not strange gait to a disease X, Y, or Z. So it's not of a disease, but it's about the sign. So do we need to come up with a disease model at all? And if so, how would it look like if we care about a sign rather than a disease?
I think it's important to see where this concept comes from. So you can provide from the literature review a disease conceptual model where you see, okay, in this sign and symptoms, we are going to focus on this. But this has been highlighted as important for patients from the literature review and from interviews of patients. So maybe you don't need to have a proper interviews of patients, because from the literature review, it's super clear that this has been highlighted, but you need the background research, you need the literature review just to show that this subcategory, these symptoms, it's actually part of the disease, and it's an important one. It's enough, it's true. And because in a trial, you don't need to assess all of them. You can just select one, because in fact, your treatment affects only one symptom, so it makes sense. But you need to show that this is actually an important concept from the background reviews. And that's it.
Okay, thank you. Then Annette Mert is asking the following: in the ataxias is not actually a monolithic disease, but a cluster of disease. There are more than 200 different ataxias, and they're quite heterogeneous, not only in terms of, and in terms of the motor component, also with the non-motor components. Her question is, how then do you define a concept of interest for a specific disease? By what is most common to all patients of a specific disease? So if you have an ataxia A, B, C, for ataxia A, you take what is most common of ataxia A and what is most common for ataxia B. So do you just take by commonness or frequency in the respective group?
Yes. So here it's true that it could be complex. Always the same thing is to show that what you're actually assessing makes sense for patient, that it's actually something that patient mentioned as important. So you can have also a conceptual model for a disease-specific subtype. That could also, even if it's one, okay, it's just symptoms, but you can have granulometry in the symptoms. What is its imbalance? Is it? It could have different facets as well. So it's fine. I mean, the conceptual model is just to show that you have performed reviews to make sure that what you assess is actually important for patients. So it could be super simple, super complex, could be focused on one type of patient, could be the broader system. As long as it shows, because then it will help you to map as well what questionnaire you're going to use.
Okay, thank you. Then Tanya clarified her question about the phenotypic conceptual model. I don't know whether it's a fair question to ask you, Céline, because doing ataxia for more than 25 years knows it's still a question I cannot answer. So I think it's not easy for you to answer, but let me try to. I think it's a crucial question which keeps coming up, and I don't have an answer to this. The point is the following: is can you come up with a conceptual model for ataxia, or do you need to have a model for a specific disease or for a specific ataxia type? And the implication for this, if you just need a model for ataxia as such, it would suffice to validate the context of ataxia as a model, and then it counts in any ataxia patient for whatever subtype. And then you don't have to go into the subtype. So I don't know whether you can take this question.
That's a good question. I think it really depends on what will be the trial that you're going to plan on having. What will be the population? If the population is a mix of different types, then yes, you need to have at some point either three, if you have three subtypes, or one together, but maybe one together is not very easy to capture. If in your trial there's just one type, do not bother having all of them. It's just, it really needs to really provide a rationale for your COA selection. I don't know if it helps to answer, but yeah.
Matthias, vielleicht maybe last question, being mindful of the time of participants, although we started a bit late. So last question, you can pick from the ones which are still answered.
I think the one from Sarah is more sub question to the earlier. We can even jump over that one.
Choice.
There is just one, maybe just to link to your last point, but I also think I think what is important always to keep in mind, what is important for the respective trial population. So meaning if you have, it doesn't matter if your ataxia is only in your symptom in your only specific subtype, also maybe in 100 other ataxias. But if it's important in your specific ataxia, it's important in your specific ataxia. And that's the concept of interest, which is important of them. And then you, at the end, you will have to show it for your trial population, and that nicely complements your answer, Céline. What you said, if your trial population is across ataxias, and the common denominator and also the target of the common denominator treatment target is, for example, gait ataxia, then it suffices if you have target gait ataxia, and that's your concept. And whether it's in 1, 2, 3, or 6, it's a common denominator of your trial population plus a common denominator of your target. And that is
absolutely. Yeah, it's exactly this. It's really, you need to support your why if your population is all the ataxia, you need to show that from the interviews or the literature review that this gait issue is reflected in all type of ataxia. If you're dealing with just one, you just
Okay, thank you.
Okay, nice final words. So thanks a lot, Céline.